Hello, everybody. Thanks for this great honor. It's been a great time so far in Sweden. And I hope this great memory will stay with me forever. I'm going to start with just a short introduction about myself. I'm, again, one of those weirdos that did interdisciplinary stuff. I was raised. Where's the laser? Here. I was raised in Cyprus, here in the Mediterranean, had a great childhood. Then moved to Germany to become a hippie and study physics and mathematics. In the meantime, spent some time in France, where I discovered my passion for biology, or applying methods from physics and mathematics in biology, and as well as the mountains. And so these two passions were the reasons I eventually moved to the west coast of Canada, in Vancouver, where I have the opportunity to pursue both of these passions. So during my PhD there, I worked a lot with molecular sequencing techniques, DNA analysis. I did a lot of mathematical modeling as well, physical modeling, and it all revolves around understanding the ecology of microbial metabolism and its role in the environment. So let me begin my scientific talk with a number. There are over 10 to the 30 bacterial cells living on Earth today. So that's 1 million times more the estimated number of stars in the universe. The total bacterial biomass on the globe, as well as the total bacterial photosynthetic activity, are actually comparable in magnitude to those of all plants combined. So we don't see them, but they're there, and they're extremely active and extremely important. For example, cyanobacteria have changed our atmosphere. They oxygenated it about 2.5 billion years ago, thereby inducing one of the biggest chemical transitions in Earth's history and making Earth habitable for most of us. And even today, bacteria and other microorganisms, such as archaea, catalyze the bulk of global biogeochemical cycles, such as nitrogen fluxes, oxygen fluxes, carbon fluxes, at ecosystem scales all the way up to global scales, and including the human gut. So understanding the role of microorganisms, especially bacteria, in the environment is not just important for environmental purposes, industrial purposes, but also for human health. And most importantly, in my opinion, it is a big representation of life. So understanding microorganisms is paramount to understanding how life interacts with the planet at geological timescales, but even today. The problem with understanding microbial systems at ecosystem scales, especially, is their incredible complexity. So, for example, this small tube of seawater can contain millions of microorganisms, so single-celled organisms, and thousands of microbial species. And all these organisms, they interact in very complex ways. For example, by exchanging byproducts of their metabolism, by fighting each other using antibiotics and things that fight antibiotics. They get infected by viruses, they attach to each other, and so on. So we're talking about a very complex interaction network, and modeling all of these processes at the same time, for example, using computer simulations to predict what's happening, is practically impossible and probably will remain impossible forever. But often, we're just interested in the net effects of all of these complex processes on overall biogeochemical fluxes. For example, fluxes of different carbon species, oxygen species, nitrogen species, and so on. And fortunately, the bulk of these fluxes is catalyzed by a core set of ancient genes. And these genes are found in many different microbial taxa. So it would be neat if somehow we could predict the dynamics of these genes at ecosystem scales. Even though genes are part of genomes and organisms, if we could somehow find principles by which we could predict the dynamics, especially the catalytic activity of these genes at ecosystem scales, then that would be quite an elegant way of understanding ecosystem scale processes. And what that means for in the context of ecological theory is that microbial systems, perhaps, hopefully, can be dissected into two complementary facets, and kind of poetically call them community metabolic function on the one hand, and taxonomic composition within functional groups on the other hand. So when I say functional groups, I mean groups of microorganisms that perform a particular metabolic reaction or a particular metabolic pathway. And the big assertion here is that these two facets, they're kind of conceptual, these two facets are somehow independent of each other or uncoupled, so that we might be able to find a theory that describes this aspect of microbial systems, and we don't have to worry about the different species representing these functions and the different processes that influence those species. And during my PhD and continuing now, and with a lot of collaborators, we've been doing a lot of work in testing these ideas, refining these ideas for different types of ecosystems. And so I'm going to tell you about some of these examples. We were particularly interested in the ocean and microorganisms in the ocean because, well, the ocean is probably the biggest mixed ecosystem on this planet. And it's important for global productivity as well as climate. And in the first attempt to test these ideas, we used sequencing data, molecular DNA sequencing data from a recent global ocean survey. It was quite handy for us. And we grouped microorganisms based on their most likely metabolic function in the ecosystem. For example, we grouped microorganisms in the ocean as oxygenic photosynthesizers, or methanogens, or methanotrophs, and so on. Functions that are relevant to ocean chemistry. And when we put all that together and we took into account the abundances of these different organisms, we found very strong patterns by which these different metabolic functions are distributed in the ocean over space and time. And here I'm just showing you, it's just one way of showing this information. We have different metabolic functions here, and the darkness of each cell is sort of the abundance of that metabolic function in the ocean, and each little column here represents one particular sample, and the samples are ordered by depth, and you can see a very strong structuring of different functions with depth. In fact, when we compared these different metabolic functional groups with environmental information, so oceanographic data, we found very strong correlations between these different metabolic functions and different environmental variables such as oxygen concentration, nitrate concentration, temperature, and so on. And that's kind of graphically represented here by big, colorful circles. And on the other hand, when we compared the species composition of these microbial communities at each space-time point with these environmental variables, we found very weak correlations. So it's represented here by weak, smaller circles. So the take-home message here is that somehow the physical-chemical environment in the ocean very strongly couples with the metabolic aspect of these microbial systems, whereas the coupling to the taxonomic makeup of these systems is very weak. It doesn't matter which species represents a particular function. It's much more relevant from an environmental point of view, which metabolisms are present and at which strength. So let me see. This is basically what I just said. In essence, we found that certain factors, including the environment, is strongly coupled to the functional structure of these microbial communities on a global scale. And something else determines which species get to represent each of these metabolic functions in the ocean. We don't know at this point what these factors are, but the fact that they are different factors means that these two aspects are in some sense actually decoupled. They are determined by different mechanisms, by different factors. So it's a neat observation, it's global, and it implies that if we somehow could control for the environment, for physical chemical variables in the environment, then in particular we should observe that this aspect of microbial systems is conserved. And even if this aspect of microbial systems is variable. It's a special case of this general paradigm. And we went and tested this prediction using another types of ecosystem. This time we looked at mini ecosystems hosted within these bromeliad plants. This is in Brazil, in a sand dune forest. And these plants have a very particular shape of leaves. And these leaves accumulate rainwater and detritus over time. And this detritus supports very rich microbial systems. So a lot of microorganisms live in this detritus. And we used DNA sequencing techniques to sort of look at many replicates of this type of ecosystem. They grow next to each other, so it's very neat. And we looked at what kind of organisms do we find there, what kind of genes do we find there associated with different types of metabolisms. And what we found was that if you count the number of, or the abundances of different gene groups associated with very important types of metabolism, it's kind of coarse-grained. Fermentation, for example, genes associated with fermentation or genes associated with oxygen respiration, and so on. The proportions of these genes in each of these ecosystems is rather conserved. So here, each column is a different version, a different copy of that ecosystem. It's a different bromeliad. And each color is one of these gene groups. And you see that all copies of this ecosystem have very similar composition in terms of the genes. And when we looked at the kind of species we find here, it's just a huge mess. So this is the same type of figure as the previous one, only here I'm showing genes and here I'm showing species. More precisely speaking, these are operational taxonomic units, so they're kind of a more practical way of defining species for microbes. But the point here is that every copy of that ecosystem had the same genetic composition, but the species are vastly different. So that speaks to this idea that we might be able to find theories and tools for predicting the genetic makeup and maybe even the activity of these ecosystems, and not worry about what kind of species are there. So it's a verification of what we predicted earlier from our ocean study. And one of the additional cool things we found using a lot of stats was that what shapes the species composition in these systems might be biological interactions between the microbes. So certain types of competition, antagonistic interactions, infection by viruses. There's reasons to believe that viruses could really change what species happen to perform every type of metabolism. And using mathematical models, actually, we find very strong support for that type of interpretation. We created simulation models for a similar system such as the bromeliads. And we found that it's actually quite plausible that viruses drive ongoing changes of bacterial lineages in those systems over time and space, even if, in this case, methanogenesis is rather conserved over time. So we have statistical evidence, surveys, we have mathematical evidence so far, and moving forward. So this is a strong kind of indication that, at least in certain cases, we could understand the dynamics of the genes regardless of which organisms happen to host these genes. So we wanted to move one step further and figure out how to do that. So I'm just going to skip that. We wanted to create actual mathematical models for real ecosystems that only consider the dynamics of genes. So it's like a gene-centric or pathway-centric view on ecosystems and microorganisms. And we did this on Saanich Inlet, which is an inlet off the coast of Vancouver Island here. And the cool thing about this inlet is that it periodically becomes oxygen depleted. So these oxygen depleted regions are quite widespread in the ocean. There's a lot of them. They're usually small compared to the overall ocean volume, but they're quite important because they select for specific types of microbial metabolism, anaerobic metabolism, including fermentation, denitrification, and other things that have a strong effect on the chemistry of the ocean, and even beyond that, climate, because they influence methane emissions from the ocean and so on. So what we did, we had access to a very rich data set. Actually, some of my collaborators have been going to this site for many years now, and I've been joining them occasionally, collecting very rich chemical data across the water column over time. So this is many years. Oxygen, nitrate, sulfite, and there's many others. At the same time, we've also been taking DNA samples. And in microbial ecology, we can use those DNA samples to quantify DNA or gene abundances, transcript abundances, protein abundances corresponding to these genes, and so on over space-time. And we created a mathematical model that tries to explain the different types of metabolisms that we were interested in from a very gene-centric point of view. So the peculiarity about this model is that it somehow treats every gene as an individual replicating entity, even though every gene is part of a genome and maybe even paired with other genes. But we just said, let's just try it. Let's just think about genes as individual entities, and they interact and they compete and so on. And it actually works. So we calibrated this model. There were some parameters that needed to be calibrated using the chemical information that we have at a particular time point, actually, when the system was rather at equilibrium. And using this calibrated model, we were then able to predict the profiles of different genes. So we have genes, for example, here involved in ammonia oxidation or nitrite oxidation and so on. And the first row is DNA, the second row is mRNA, and the third row is proteins, all corresponding to the same genes. And colored curves are model predictions, and the gray dots are our actual data. And you see that we're able to actually explain quite a bit the depth profiles of those genes, both DNA, mRNA, proteins. And so it's quite encouraging, although, I mean, it's just one example, and we don't really know how widely applicable these types of models are to other ecosystems or more complex settings. In particular, this model was applied to steady state situation. So when the oxygen depleted zone in this inlet was rather stabilized. So we don't really know how to extend these types of mathematical models to more dynamical settings where things change over time. And in fact, Saanich Inlet is subject to intense fluctuations over the year. So what we're doing now, I'm just going to give you a very short glimpse to what we're working on now. We have the chemical data over many years and depth, and we're trying to reconstruct fluxes, both spatially and temporally resolved. So fluxes of different chemicals. We're using inverse linear transport modeling. It's a technique borrowed from other domains of physics. We then estimate metabolic reaction rates, again, ecosystem scale over multiple years. We use then certain mathematical models to predict what the densities of different genes involved with this reaction should be. And then eventually we're comparing that with qPCR-based cell counts and different sequencing data that we have. So this is work in progress, but we're hoping that one day we'll be able to have a framework that combines geochemical information with molecular information and things that we know about how biomass is produced and how genes proliferate, all under one hood. So send me an email in 2 years from now. I'll let you know what happens. So the big picture, to finish with, I would like to just kind of share with you how I think this all fits together in a worldview. The question that I think we're beginning to scratch on here is the following, namely, at what organizational level should we be describing life in order to understand how it shapes ecosystems and Earth? Do we only need to think about genes? Do we need to think about higher order structures, higher order forms of organization, cells, colonies, organisms, and so on? And the information I've shown you today suggests that we can actually learn quite a big deal about how life shapes the world by really only looking at genes. Every model is wrong, but some models are useful, and this model seems to be quite useful. So with that, I'd like to thank everyone, SciLife Lab and Science, and all my collaborators. Thanks.